I'd like to introduce Dr. Lorenzo Ferri. I think if I were to read all of this, it would be longer than his presentation because he's done so much work. But I will read just a couple of sentences. So Dr. Lorenzo Ferri is the David S. Mulder Chair in Surgery, a clinical scientist in the departments of surgery and oncology, specializing in the management of complex malignancies of the foregut, and heads the McGill University program in upper GI cancer. The rest you can read. I made I pushed my way into Dr. Lorenzo's world a couple of years ago, and he might be regretting it. I've worked with him on a grant prior to the one that we've just begun now, and I'm really, really excited about this one as well, although I don't understand it yet fully. So, yes. So, Dr. Lorenzo Ferri.
Thank you so much. Does this microphone work? Yeah, it does. Okay, so I'm going to talk a little bit about some work that that my lab's been doing for the past maybe 8 to 10 years, and how we're trying to really challenge the way we're treating all cancers, but the cancer that I treat primarily. But it gives you sort of a framework of the concept of treatment of any disease and how we can do better. I am a surgeon, so I come from a little bit of a surgical perspective, but I feel that our advances in this disease, in stomach and esophagus adenocarcinoma, are going to come not from surgical innovations, but more from innovations of systemic therapies. So I'm going to get one of these. One button's going to work eventually. Okay, so this is the beast that we're trying to treat. It's called Gastro-Esophageal Adenocarcinoma. It is now, we have good data to show that this disease, stomach and esophagus adenocarcinoma, are very similar, and they should be treated as such. So we know that esophagectomy or operations provide the best chance of cure from when you have a localized or locally advanced cancer of the stomach and the esophagus. However, even despite our best efforts, most patients will have, a significant portion of patients undergoing surgery will develop metastases or cancer that comes back, like this gentleman that I operated on not too long ago with a metastasis to the liver. So we need some help. Surgery alone is not going to solve the equation, not going to solve the problem in the vast majority of patients. We need some other form of treatment to help with this. And that really gives you the concept of chemotherapy. We need to have treatment that goes everywhere around the body, it tackles the cancer wherever it may be hiding, and we need to maximize our ability to cover the tumor, those small cells that could be hiding elsewhere. And so there are a couple of treatments for this disease, but the one that's held the most in North America and Europe is giving chemotherapy before and after surgery. So the goals of the chemotherapy is to improve systemic control throughout the entire body. It leaves the tumor localized for surgery or another treatment like radiation therapy. And there's been a bunch of trials in this. The first trial that really made a difference was called the MAGIC trial. This is the only survival curves I'm going to show. The MAGIC trial out of the UK, which showed this epirubicin-based chemotherapy as the best treatment. We and others showed that after this, that docetaxel or Taxotere seemed to be really effective in this disease. It was the first trial that I ran when I first came on staff in McGill. And subsequent to this, a trial comparing ECF or epirubicin-based to a docetaxel-based treatment has really shown the benefit of our current FLOT-based chemotherapy, which is really a docetaxel-based triplet regimen. And that's sort of the standard bearer for using chemotherapy around the time of surgery. And so this is the current standard of care in most of North America. Now, we've also shown that even though it's for stomach, and their initial trial showed that it was really good for stomach and esophageal gastric junction cancers, one of the criticisms that for esophageal adenocarcinomas, we didn't really have really data. So In a non-randomized trial, we looked at our data with Trinity College Dublin and Newcastle, two other large high-volume centers, and showed that it is also very effective for this disease. And based on the trials from FLOT and additional studies since then, this has led the docetaxel or Taxotere, the same drug, base triplets, is the standard of care for neoadjuvant or perioperative chemotherapy for both gastric and esophageal adenocarcinoma. So that's the basis. That's the best thing we have going right now. So most patients that we see, this is who have localized or locally advanced, that don't have spread elsewhere, but just to the regional lymph nodes. This is the current treatment algorithm that we have. We have someone that is diagnosed as like this gentleman I treated maybe 5 or 6 years ago. He has a tumor in the bottom of the esophagus extended into the stomach. It's adenocarcinoma. It has no spread anywhere else except for some lymph nodes around the tumor. We give that patient docetaxel or FLOT-based chemotherapy. We give usually 4 cycles prior to surgery. At the time of surgery, take out that tumor, and even if they had a poor response, that tumor had no benefit, did not shrink at all. You know what our current standard of care is? We continue on with 4 more cycles. And so we're just banging our head against the wall here in a lot of these patients, because that's a standard of care. If we change it, there's no data to say that changing makes it any better. So right now, this is the treatment we give, even if they did not respond to the 4 cycles of chemotherapy we gave beforehand. So this benefits some patients, but a large number of patients do not benefit from this blind approach to patient care. And why is that? Because a lot of people, there's a certain number of patients, around 40% of people in stomach and esophagus cancers are innately resistant to this type of chemotherapy. It's pretty effective, but a lot of people don't have benefit from that. Their tumors don't respond, like this gentleman's tumor did not respond. And additionally, around half of those patients who do respond will have developed an acquired resistance to that chemotherapy. So giving 4 more cycles after we've already given chemotherapy doesn't make much sense. So how can we make it better for our patients? So this sort of highlights 2 different scenarios. 2 patients are treated in the past year. Top is a 74-year-old woman who's MSI high. We heard a little bit about that today. At the time, we did not have any trials available for immunotherapy, but you can see a very large tumor. I can't point at all, but a very large tumor on the scope, and below that, the PET scan. You see the big white thing is a tumor. So we gave that patient chemotherapy. In the middle after 2 cycles, you see is a good response on the scope. And after treatment, it almost nearly disappears, that chemotherapy. So this patient responded really well to that chemotherapy, that FLOT. She's a good responder. The tumor is highly sensitive to that treatment. It makes sense to continue treating that patient with the same chemotherapy. Conversely, there's another gentleman I treated maybe 3 years ago. He has an adenocarcinoma of the esophagus in a field of Barrett's from reflux. You can see a small tumor right there, actually 2 small tumors in this gentleman. He received chemotherapy. And in the mid-treatment box, you see a little bit of ulceration in the middle. But after chemotherapy, it's actually gone bigger. And now he's actually on PET scan. Right before which we planned to go surgery, we found that he had progressed in the liver. So this gentleman had no benefit at all and actually was harmed by that chemotherapy. His tumor is not responsive. That tumor has poorly responded to that tumor. So not only did he not benefit from that chemotherapy, he got all the toxicity of 4 cycles of pretty tough chemotherapy that was basically futile. He lost 3 months of his life where he could be getting a better treatment. So he's lost on all these different factors, all these different areas where we could have been better. We failed that patient. So these are the challenges that we have. We see this every day in the clinic. How do we identify? How do we find the patients who are going to respond well to chemotherapy? How do we identify those who will not respond and come up with new treatments for those patients? So these are challenges that we've recognized that we had to overcome. So how do we predict if a patient's tumor will respond to the treatment? How do we identify new biomarkers? We heard a little bit of that from Dr. Klemper's talk. The sort of proteins in the tumor, can we identify new ones that will let us know what treatment we can give? We have 2 of them right now. We heard a little bit about HER2 and PD-L1 and MSI high, how that may predict how someone's going to respond, but not everyone has those. So there are new biomarkers that you can identify who's going to respond to key treatment. And what are the factors that are driving chemotherapy resistance, either innately, initially, or acquired? How do we understand them so we can design new drugs to treat the patients who have those mechanisms that are driving their tumors? And furthermore, what can we do to overcome therapy resistance? If we know we have someone who has a resistant tumor, what drug can we use to salvage that tumor from being treated? So based on that, and based on some work that we've already been doing on this trial, we got together a large group of clinicians and scientists and patient advocates, and we came up with this as the grant that we just received, this Overcoming Therapy Resistance and Esophageal Gastric Adenocarcinoma Trans-Canada initiative called the Treat Canada. I spent probably as much time on the title as I did on the grant. I like the Treat Canada. An overarching goal of this project was it was building on a grant that I already had from the Department of Defense of the U.S. as well as another one from the UK. So we'd already had some research, some data to work with. But our overarching goal was to identify and improve therapies for people with gastro-esophageal adenocarcinoma. Understand the molecular drivers of therapy resistance from a multi-omic perspective. I will just explain what that means. Avoid ineffective and toxic treatments of patients who will not respond. Identify alternative treatment strategies, approaches to salvage resistant tumors. So that was the ultimate goal that we've been tasked by the Kidney Cancer Society and given a large amount of money to try to come up to a plan to treating patients better. So we assigned this as our first all-team meeting, and we combined a multidisciplinary group of patient advocacies, cancer cell biologists, computational biologists, bioengineering, clinicians, genome scientists, all together so we can speak on the same tackle the cancer from multiple different areas. And we realized we can't do that all ourselves. The science has gone so much bigger. We have a lot of collaborators from around North America. It's based in Montreal, but we have an incredible group of clinical scientists across the country from coast to coast. We rely on assistance from in computational biology from Stanford, UCSD, Seattle, and a large bioengineering group from Harvard, and well as our patient advocates really drive and remind us why we're doing this work. So this team, we've built this grant on a sort of four work package platform. And it's all based on patients participating as they're undergoing neoadjuvant therapy. So this is what happens. Patients get newly diagnosed. We do a biopsy at that time. Halfway through that first treatment, they do another biopsy. And then when we get to surgery, we take out the tumor and we analyze that tumor for a few things. First things we do is we've developed a way to really grow those cells outside the body and keep them alive so we can retest drugs on them. I'll show you how that works. Secondly, we really analyze that tumor from multiple different angles. We looked at what mutations are in that cancer. We looked at what cells are what the signals that they're expressing, and we're also looking at the proteins that those cancer cells are making. And we're trying to integrate all that data to understand patients who are responding, just like that first case, and compare those to patients who are not responding, like the second patient we had. So we take that information from those two work packages and we combine them. We combine them by taking the information about the cancer and those cancer cells that we've grown outside the body, and we put them together and generate novel, clinically relevant cancer model systems. And we try to use those to repurpose drugs. It's important to recognize we have great, to develop a new cancer drug takes multiple years, even a decade or two. So if we can find a drug that works for lupus and repurpose it for a patient's cancer based on a computational pipeline, or repurposing pipeline, this machine learning pipeline, then we can save a lot of time for that patient. So we do it in 3 different ways, and I'll explain a little bit more later on. We use those cancer cells, and we do some drug testing based on the mutations they have or the proteins they express. We also do high throughput drug screening. It doesn't make a difference what mutation they have. We just test every drug that's FDA approved. We try to merge different cancer cells, like the lymphoid cells, immune cells, and the cancer cells together to understand immune therapy. And then finally, what we're really trying to do is put everything together on an organ chip, and I'll explain that a little bit later on. Finally, work package 4, we're trying to validate from this information that we get from work packages 2 and work packages 3, we're going to come up with a whole bunch of proteins that are associated with response. So we will be able to determine have new biomarkers to determine whether or not someone's going to be truly going to respond to the chemotherapy that we propose to them. And if not, what might they respond to? So this is a whole series of, and this is going to be patients from across Canada who are going to entrust us with their tumors and their information in order to be able to hopefully benefit patients from across the world. So at the end of this, we really want to challenge traditional treatment of gastroesophageal cancer. So in traditional care, we just give standard of care. That's what we do. That's where we are right now, where we just give standard of care cytotoxic chemotherapy drugs that have been around for a while. A lot of them have been around for 20, 30 years. And then in precision oncology, we have just started to enter the era of precision oncology that Dr. Klemmer talked about. And we don't have too many targets just yet. In fact, unfortunately for Gastro-Esophageal adenocarcinoma, we have very limited in our targets, and not very many patients have it. And you can also do foundation medicine. You can send your tumor off, and you get some information. That gives you a laundry list of drugs that may or may not work in your tumor. But unfortunately, even with our current iteration of precision oncology, it's still a trial and error approach to patient care. You may or may not. So you may get a drug that has no effect in your cancer. So can't we be smarter about this? Can we have a better way of treating these cancers? Maybe let's just take a feather out of the cap of microbiology. For almost 100 years, if you have an infection, you spit in a cup, if you have a suspected pneumonia, you cough it up in a cup. We grow that in a culture system outside. Then we test a whole bunch of antibiotics on it and find out which one works. The one on the top right seems to be working better, so we'll give you that antibiotic. How come we can't do this for cancer? This is the concept of functional precision oncology. So you determine exactly outside the body what is working in that cancer and find what to be able to give to the patients. We've been doing this for 100 years in microbiology. We should be able to do it in cancer. What was the problem beforehand? We're missing the adequate cancer model systems. So this gives me the concept of what's called a patient-derived organoid. Over the past 10 years, pretty much, we've developed new ways of growing cancer cells outside the body called these organoids. They're three-dimensional cell cultures. They're self-organizing. They have stem cells in there, which means they can create all different cells in an organ. And they maintain the heterogeneity. Like a tumor is not just one tumor. It's a whole bunch of different sort of cousins, as opposed to all one. They're not all twins. And so if you treat one cousin, they may not may not have the same impact on another cousin. So the whole idea about these organoids, they maintain all those different cell types. So over the past 10 years, we've really invested a lot of resources. So this was first developed, and this technology was first developed in Netherlands. We were one of a really very early adopter. So over the past 8 years, we put a lot of resources into developing this concept here in Canada. Currently we're around 400 patients where we have developed these organoids. So we take out a tumor from a biopsy, we grow them outside the body in these organoids, and we also take some of the immune cells and the structure cells, and we grow them as well. And we merge that information with the sequencing data I talked about earlier. So we use those organoids. And we've shown that those organoids, they look exactly like the cancer from which they came from. So here on the left, the actual tumor is a third row. You have the organoids as a middle row. They look exactly like histologically under the microscope as the tumor from which they originated. You can see that quite nicely in the top right hand, where you have the tumor on the left, organoid on the right. Looks exactly the same. And genomically, it maintains the genome largely from the tumor from which it came. So the question is, if it looks like it and it's genomically similar, does it act the same way? And indeed it does. So these are examples of organoids that were generated from a patient prior to chemotherapy. That patient received chemotherapy, and the patients, we either have a tumor from the organoid from a tumor that responded really well to chemotherapy in the patient. We can replicate that in the organoids. Can you do the video for the first one, please? So the top left is a patient who responded a really good response, and you can see After 3 days of chemotherapy in the Petri dish, it explodes. It's like the Death Star, from Star Wars. It's absolutely destroyed that cancer, showing this and that's represented in one of the green, the green kill curves on the right. Whereas you can click the next one. This is the one who did not respond. After 3 days of chemotherapy in the Petri dish, these organoids have not changed at all. It looks like it's been tickled. It's not being killed at all, and that represents the red ones. So our challenge right now, how do we turn those red ones, those ones who are not responding, how do we make them respond? So if we identify outside the body that it's not going to respond, so first thing we know, we shouldn't give that chemotherapy to that patient. Secondly, the next question is, what do we do to make sure it responds so we find the right treatment for you? So that's where we take some of that information we got from those sequencing data. So I've taken those, at the same time we generate those organoids, we're also doing a whole bunch of sequencing. We're looking at what mutations are driving that cancer. This is an example of one. It's a lot of complex. I barely understand this myself, and these are my PhD students. They're a hell of a lot better than I do. From our sequencing, our single cell sequencing, we identify a cluster of cancer cells, and they're expressing this protein. They're expressing the genetic material in order to generate this protein called CDK12. And this is an example for one patient. We don't do this for every patient. We do this for a single patient. Now, we look at the organoids that we generate from that patient, and we can see there's a lot of CDK12 expressed in that organoid. That means maybe it responds to a chemotherapy or a drug that targets that CDK. And indeed, it does. You can see here on the SR4835, the lower that curve is, the more response it has to that treatment, as opposed to DCF, which is a standard chemotherapy that the patient got, doesn't respond at all. So what if you combine that with DCF? We find that there's actually synergy. They work together. So you look at the bottom blue, the green line. It starts to kill more and more of those cancer cells by adding this drug that we would not have thought of otherwise had we not had the sequencing data. And another way to do it is, who cares about what mutations are? We just want a drug that works. We want a drug that's tolerable and that works, irrespective of what genome is, what mutation is driving that cancer. So another way to look at it is to just go backwards. Let's try something, go top down. We take those organoids and we throw every drug that we have at it. So this one, there's a few libraries out there. This is over 3,000 marketed drugs are FDA approved. Covers everything from cancer drugs to lupus drugs, from arthritis drugs to skin drugs. And we use these and we put those all together on the organoids. It's an example of what it looks like on a robotic platform here, where we take those patients as an individual patient's organoid. We grow it out after 2 or 3 weeks. We can do this test where we test all these drugs to see which one's going to work. First, we give the media. Then we put the organoids in there. Then afterwards, we take the media and make sure that they continue to grow. And after we've done that, we do this. This is way too much work for a student to go pipetting here. It'll take them weeks. Here we can do this in hours, and we get all the drugs. So each patient has 10 of those plates. That's 3,000 drugs we're able to test. And then we see which one works. So out of 3,000, this is an example of a study where we took 3 organoids from tumors of patients who did not respond to chemotherapy. And we found out there's around 200 drugs where they had some function in the role. And then we looked at the ones that worked in all 3 of them, which means there might be some commonalities we can look at. We came down to 20 chemotherapy compounds that worked in all 3 of those factors. Now, maybe some of them are too toxic to use. Maybe some are we're not able to. We need to hone down a little bit further from there. So how do we do that? So here you can see all the different sort of pathways that are explained by those 20 drugs. And we look at those a little bit closer. Maybe we can hone down a little bit further from those 20 drugs to maybe 5 drugs. So what we do, we take the data we got from the sequencing data, we integrate it from the high throughput drug screen data and put that stuff together. One example is the insulin growth factor 1 receptor. So that's something we would never have thought about otherwise. So we look at it at the sequencing data, and indeed, it's the poor responders have more IGF-1R expression, and you look at the single-cell transcriptoma data, all those red spots on the gray blot are ones that are poor responding. And then we look at that one little focus on 14, that has a lot of the high IGFR1R expression. So maybe we take that data from the high throughput drug screen with a robot, we get the sequence data, merge it together, and we look at IGFR1 inhibitor and show that it has really synergistic with the chemotherapy. So it may be a combination. That's a combination that we should use on the patient. So there are limits to the organoids, however. As you can see here, there's a crossover in the middle there, where it doesn't separate the poor responders from the high responders. Why is that? Because organoids are just one component of the tumor. Dr. Klempner mentioned, there's all these other factors like the blood vessels and the immune cells and stromal cells. So those are missing. Secondly, it's in static in vitro. Our body is full of vessels where the heart's pumping the treatment chemotherapy through your veins, and it goes through those structure cells and then gets to the cancer. We can't replicate that. The tumor cells are just bathed in the chemotherapy. So how do we address all those factors? And we know that the tumor immune microenvironment really influences cancer biology. So it's not just the cancer cells, it's the other structures that are in there like immune cells, fibroblasts, endothelial cells. And we know that those fibroblasts, they can impact how chemotherapy responds. So it would be really important to put them all together to get a really better understanding. Once we've honed down to maybe 5 or 6 drugs, we need a new model to tell us which one of those 5 is actually going to work in the patient. And that's where I'm really excited about this work that we're doing now, which is a human cancer chip. This is it's a microfluidic device where you take it, that's the thing on the right is the size of a USB key, even though we don't use them anymore, basically the size of your thumb, and you try to recreate all those factors in a regular organ, the blood flow, the stretch. Every time you take a breath, you're stretching your lung cells. Every time you swallow, you have peristalsis in the esophagus pushing down food, and that stretches those cells, and that impacts tumor biology. So what we're doing, we take those organoids that we've generated, and we recreate that organ in these chips. That's what's really exciting. We take the organoids, we put them in a top channel, and we take the fibroblasts and the immune cells, we put them in the vascular channel, so they're separated by a membrane, just like a normal tissue, a normal organ is. So once we're able to do this, you can see once we've done this, we can really, right now we're at 2 cells, and we're trying to get to 4 cells, all different cell types. So we basically take out a tumor, squeeze it all out, and then we're try to recreate that organ from where the tumor came back in this organ chip so we can understand which chemotherapy is going to work best. This is what it looks like afterwards. You can see a normal esophagus on the left and a esophagus cancer chip on the right. You can see the bottom right, in the black and the top right here, this big tumor that has grown out on that organ chip. So it really replicates what it looks like in the normal tumor. In fact, if you take do histology on the primary tumor and on the esophageal adenocarcinoma chip, it's exactly the same. You give this to a pathologist, they'll say these are the exact same tumors. So we've really been able to recreate what it looks like in the actual normal tissue in this chip, that we can test those drugs better. So this is the concept. We've developed organ chips for multiple patients. We've done maybe 20 right now, and we've compared patients who are chemosensitive, whose tumors are sensitive to that tumor. Can we recreate that in the chip? And we compare them to chemoresistant tumors. So you can see here, on the top left, you have a chemosensitive tumor. On the top, just to the right of it, you have a chemoresistant tumor, and they have both immunofluorescence and electron microscopy. If we give chemotherapy, the same chemotherapy the patient got through that lower channel at the same concentrations that the patient got, we can recreate entirely the exact same thing we see in the patient. In the bottom left, you see a treated cancer. All those cells on the top, all those light blue teal cells, almost all of them are gone. That's after only 1 cycle. You can see that membrane is almost completely destroyed. Just to the right of that, you see it's reduced a little bit in the chemoresistant, but they're having maintenance of a layer. And you see the electron microscopy, the third in the bottom, you see all those cells are destroyed. They're exploding, whereas the chemoresistant hasn't been touched by it. So we've been able to show that we can really recreate what happens in the patient in this organ chip. The concept is to take those drugs that we've identified from the high throughput drug screens and the variants from the organoids, which we can do 3,000 at the same time. come down to 5 to 10 drugs, tested on the organ chip, and confirm which one's actually going to work in you. So that's what we're trying to do right now. There's been a lot of excitement about this, about using functional precision oncology. There's been a few studies. It started off in Utrecht in Netherlands. So there's one trial that they've published so far, which did not show that it was conceptually possible, but there are issues. It took too long at their center. It took 10 to 12 weeks. That's too long. That doesn't make any sense for someone who has to have chemotherapy soon. You can't wait 3 months or else it's too late. But the concept is there. So what we're really trying to do at the end of this grant is to provide functional precision oncology to Canadian patients with this disease on a clinically relevant timeline, a timeline that makes sense to you, to patients across Canada. So this is what we have right now. We spent a lot of time to build this up, where we're from a biopsy, initial biopsy, we're able to grow the organoids within around 3 weeks. We can test them a week to 2 later, and we have the results of our high-throughput drug screen 3 days after that. We then integrate the information from the genomics and transcriptomics, because we get those results by 3 to 4 weeks afterwards, and merge. You come out from that, you get a short list of 4 to 5 drugs that you validate on the esophagus organ chip, and you know which exactly one works. So our concept is to have a platform for functional precision oncology and esophageal cancers that not only tests systemic cytotoxic chemotherapy, the standard chemotherapy, because it does work in a lot of patients, which ones work, but you want to know in advance if it's going to work on someone before you give it to it. We can also test biologics, antibodies, and inhibitors. Antibodies a little bit harder, but definitely inhibitors, and definitely immunotherapy, because we are starting to grow flow immune cells through that lower channel. So that is our concept. Take this high throughput patient-derived organoid, give us an idea, get it to our low throughput, but extremely high fidelity organ chip to give an idea and give the patients at least some confidence that what they're going to get is actually going to work in them. This is used to be a pipe dream. I do think in the course of our trial, we should be able to get to the stage. So what is esophageal? What is the treatment in the future looks like for gastro-esophageal adenocarcinoma? So instead of just blindly giving 4 cycles of chemotherapy, could we identify biomarkers of people who are going to respond? Can we do this whole process beforehand? We're not there yet, because it still takes around 6 weeks to get that whole process through. But maybe we'll then identify biomarkers of those who will respond to it or not. Maybe where I do think it's going to make a difference is in the adjuvant setting. For those patients who did not respond, patients who respond to that chemotherapy, they're well served by the chemotherapy. Let's continue. It's done its job, just like that first patient I presented. For the second patient, why continue banging your head against the wall and having that poor patient suffer through treatment's not working? And that's an area where we can tailor the treatment based on organoids and organ chip, so we know what's going to work in that patient. Thank you very much. As obviously, I did none of this work. This is all done by my students, and it's cost a lot of money to do this stuff. So we're very grateful to all our donors, our patient donors, as well as this is the sources of funding we have. Thank you very much.